Thank you for attending our bi-weekly MSQ CEO webinar series. We're excited to host Aruna Bio and both its CEO and CSO, Chief Scientific Officer, co-founder Stephen Fromm and Dr. Steve Seiss, respectively. My name is Mark Estigarribia, Managing Director of MSQ, a New York-based cross-border advisory firm focused on global biopharma transactions. We are honored to introduce Aruna Bio. Aruna is unlocking new possibilities for millions with neurological disease by developing their own proprietary neural exosome platform. Aruna Bio has succeeded in gaining access across the blood-brain barrier. We are looking forward to learning more about AB126, which is ideal for the treatment of acute neural conditions and chronic neurodegenerative diseases, which could transform the lives of millions of people around the world. Here today, we have Stephen Fromm, Chief Executive Officer of Aruna Bio. He was former President, CEO, Chairman of iGate Pharma, who successfully took iGate public and completed multiple M&As and licensing deals. Prior to iGate, he was the CFO of Centelon, a subsidiary of Sanofi Aventis. Also at B of A, he was Director of Global Healthcare Corporate Investment Banking Group and headed up European Life Sciences. Also with us is Dr. Steve Seiss, Chief Scientific Officer and co-founder of Aruna Bio. In this role, he directs the company's research operations. He is also a University of Georgia D.W. Brooks Distinguished Professor and the Director of Regenerative Bioscience Center. Steve holds a Georgia Research Alliance Eminent Scholar Endowed Chair. Prior to that, he was at Advanced Cell Technology, the first company to advance human clinical trials using human pluripotent stem cells. Steve, before we start, can you say a few words about your company's history, drug development, discovery to date, and how the two of you got together on this amazing journey?
Thanks, Mark. And I'm happy to take that first question about how we got started as a co-founder of the company. As you mentioned in my introduction, I've got a lot of experience in the area of stem cell and particularly neural stem cells. And I've always been intrigued over the years with the idea that neural stem cells could pack a real punch for therapeutic opportunities. However, mechanism of action and more importantly, the route of administration really inhibits the use of stem cells for many neurological diseases. It was about 6 years ago that we came upon the idea that maybe it's not the stem cells themselves, but actually what the stem cells produce, these neural stem cells that we have in our culture system. And so what they produce, and you'll hear more of this from Stephen, are these neural exosomes. So the route of administration now is that we collect those exosomes and can inject them into a neurodegenerative model or injury model and have really dramatic effects without having to do any dramatic invasion of the brain. So that's really when the company got started. We realized 6 years ago, and we were throwing away the best part of the stem cells, and that's what the stem cells secrete into the culture media. And we started with the initial animal models and progressed through many more and are progressing to the clinics today. Stephen?
Thank you, Mark, for inviting us to this, but I think Steve gave a good overview. So just so everybody understands, my journey here at the company just started a year ago. In fact, I just had my 12-month anniversary 2 days ago with Aruna. And it's been exciting. We've done a lot in the last 12 months, and we're happy to share that with everyone.
Great, thank you. Looking forward to the next 3 to 5 years, what breakthroughs do you think exosome therapy will make in the treatment of neurological diseases?
Steve, do you want to take that one as well?
Sure, I'll take that as well. Initially, I think we're right at the cusp of moving into human clinical trials with these exosomes. So I think the real stellar advances are going to be made in the clinics using these exosomes to treat neurological disease and injury. The exosomes have millions of years of evolutionary pressure to make them very useful for a number of injuries these are naturally occurring or native exosomes in the brain that can have great effects. So I don't see us being able to improve on the native form of exosomes tremendously, but how we use them in the future in the clinics, as well as moving on to different indications, are really going to be important advances. And I fully expect them to have a great impact on human health care in the future.
Thank you. So I guess this kind of goes into this question a little bit too. Although the research on exosomes is gradually increasing, it's more concentrated on basic research. Maybe you can elaborate there, but how do you view the relationship between the basic research and clinical transformation? Where are we?
Well, I think that's what we're going to talk about today, is that we're taking it from the lab and taking it into the clinic to be able to treat patients with the product. And there are a lot of things that need to come together to be able to do that. Because as you're mentioning, it's a nascent area. It's really new. And when we think about some of the more mature companies, there's only a few that have gone into the clinic, but they're using what I call more generic cell lines. And it all starts with the cell line you're using. The one advantage that we have over some of these other companies is that we're using a proprietary cell line. So you can't get access to our cell line, it's ours. And when you're using a generic cell line or a general cell line, other people have access to it. And so it's not so much the exosome that you're collecting from that cell line, it's what you're able to do with the exosome. And what Steve has mentioned is the cell line we're starting with, it's able to produce a therapeutic exosome without any modification or engineering of that cell or of that exosome. So we're excited to get this into the clinic.
Great, excellent. I guess one more question before we get into your presentation. What are your, and this is more in the, I guess, the corporate business global strategy for Aruna. What are your global expansion aspirations? Are you open to global regional collaborations? What's your strategy here?
So that's a good question, because we're a private company and we're a small company, so we have limited resources. So our main focus right now is showing that we can do the work required to take our product, again, from research into the clinic. And so there's a lot of effort around that, and it's our main effort today. So this is a product that could be used by anybody around the world, but we don't have the ability, the expertise, or the resources to take it outside of the US. And what we'd be looking for is a partner that actually has that expertise. And so coming to the market with somebody that already has exposure there, and in fact, take it one step further, somebody that has exposure and experience taking stuff like this into the clinic to treat, whether it be stroke, TBI, or chronic neurodegenerative diseases, it'd be great to have a partner that has that expertise.
Great, thank you. I just shared the screen with you, Stephen, so please go right ahead and start with your presentation.
All right. All right, are you able to see that?
Yes, perfectly. Thank you.
All right. So I'll move it forward. So just starting at the beginning here, some corporate highlights. So as Steve mentioned, we're using a proprietary neural stem cell, but it's not the stem cell we're taking into the clinic. It's actually what the stem cell are producing or excreting, and they're called exosomes. So we're collecting and harvesting these exosomes from the stem cells that we grow. Our main product right now that we're taking into the clinic, we refer to as AB126. And it has the natural ability to cross the blood-brain barrier. It also has multiple modalities, can act as an anti-inflammatory, a neuroprotectant, and it has the ability to facilitate tissue repair with its regenerative properties, okay? It's really important to understand that when you go into the CNS, whether it be for acute or chronic, they're all difficult areas. They're difficult diseases to develop a product for. And we're going after one of the ones where there's a real opportunity here if you have something that can work, especially for the patient population where you're unable to reperfuse them. All right, our first IND, we're anticipating Q3, early Q3 to file that and get into the clinic with what we call a phase 1B safety study in stroke patients. There's a real medical need for these stroke patients. And because it's acute, it's only a 90-day primary endpoint. And the beauty of this is we're the only company that we're aware of in the world that has been able to validate how our product works in an extreme stroke environment through a pig model. And it's a validated porcine model that was done at the University of Georgia. All right, having that available to us and that data and how robust it was gave the NIH the ability to award us over $5 million to make sure our product gets an IND file. And it was that data from that pig study that really did it. This product, because of the way it works, it's also something that can be applicable to all of the chronic conditions, okay? And because, again, it's a new area, there isn't a lot of expertise on how to make these things, so we have our own GMP facility. So when we think about the neural space and how our product works, it's really interesting to understand that regardless of what's causing the problem, whether it be a clot for ischemic stroke or whether it be some form of protein gone awry, they all have something in common, the acute and chronic, all right? They have an inflammatory response and they have cell death. And so the way our product works, we can have an agnostic approach. We don't care what's causing the issue because we're able to modulate that immune response and we're able to facilitate tissue repair. So it's really important to understand if we can get across the blood-brain barrier, which we do naturally, we can then go after any of these conditions. And it's important to understand too that all of these conditions, any product that's out there right now has only an incremental benefit. And we've seen that lately, whether it be ALS or Alzheimer's, the benefit is very incremental. All right, so getting to our exosome and what it's able to do, we already mentioned that it can naturally cross the blood-brain barrier, but it has these different modalities. All right, so we're able to not actually just act as an anti-inflammatory in the neural space, but also systemically, which is really important because there are a lot of these T cells or white blood cells that get trafficked to the brain after an injury or after there's a problem going on. We promote neuroprotection and we do the facilitation of tissue repair through neurostimulation. So making sure we're all on the same page, what is an exosome? So these are little nanoparticles. They're made naturally by a cell. Almost every cell in our body produces or excretes them, and it does it so it can communicate and regulate with other cells. All right, so the cell that's making them, it's going to package them up with molecules, and it's going to actually put proteins on the exterior surface so that it can actually go to other cells of interest. And then once it's taken up by that cell of interest, it's going to come unraveled, and then the molecules inside it are going to be able to trigger a response. Okay, so you typically see this when there's a problem going on in the tissue. All right, it's going to excrete a molecule or an exosome with molecules that it can communicate with other cells that there's a problem and help regulate that problem. It's really important to understand then the cell that's producing or making the exosome, it's going to have a fingerprint on that exosome based on the cell making it and the environment that is going on where that cell is making it. So what an exosome coming from a different type of tissue or cell line is going to look a lot different than one coming from a neural cell line. Okay, and that's where we come from, is this proprietary neural cell line. It's important to understand, though, if you don't cross that blood-brain barrier, it doesn't matter how good of a therapeutic you are, you're not going to have the opportunity to use it. So we've got a lot of data showing we cross the blood-brain barrier. This is just one slide of an example. And so the question that comes up, is it all exosomes that can cross the blood-brain barrier? So we did some work where we harvested exosomes from other cell lines. And so what you see on that bar graph is it's not just an exosome, it's an exosome coming from our neural stem cell line. Okay, and we were able to demonstrate that. In this case, it was HEC293 cells. And then on the left there, we have an imaging diagram that shows we penetrate throughout the whole brain and deep into the center, the basal ganglia and other regions. Okay, so we cross the blood-brain barrier. What makes us think we're going to be a therapeutic? One of the main things we look for inside the exosome are the different nucleic acids, in this case, miRNAs. All right, so we did a search with over 750 mRNAs looking for ones that were really upregulated. And the ones that we found, especially the top 12, they play a huge role in these 3 different modalities. And in fact, within each modality, there's going to be multiple mechanisms of action on how they work. So it's a very complex product we have, but it's important to know that with a complex product, you can treat complex diseases. And I think that's one of the reasons why we've seen recently for ALS and even Alzheimer's, the drugs that are out there that are getting approved only work on one or two mechanisms of action. And so the benefit to the patient is very, very small. So you need to have this multimodality approach, a very complex approach for complex diseases. All right, so we're starting with stroke and TBI. We've begun doing work in animal models for the chronic conditions. When you think about the chronic area, there's two main categories, right? The motor symptom diseases and the cognitive symptom diseases. And there's a lot of overlap between the two. But we chose two different diseases from each of these categories that make a lot of sense for our product to start with. So on the motor symptom side, ALS, and on the cognitive symptom side, we're starting with vascular dementia. But once we figure out how to dose for these two conditions, we can open up that pipeline and really expand it to other chronic neurodegenerative diseases. Here's a snapshot right now of what it looks like, our current pipeline over the next couple of years. We're looking to file the IND, as I've mentioned multiple times, early Q3. That'll get us open for the acute conditions. We're going to run our phase 1B safety study in stroke patients. When that completes, we'll do multiple proof of concept studies with different patient populations for stroke and TBI. And again, as I mentioned, we're doing the animal models right now for ALS and vascular dementia. We should have the first data coming in for ALS right around the time we file the IND for the acute conditions. But the goal is by the time we finished our acute safety study, we're filing the IND for chronic. It's all the same exosome. Again, they're not modified exosomes. They're just the native exosomes that we harvest from our proprietary cell line. And so that second IND for chronic will be a much lighter and easier IND to file because it's really going to have chronic tox data in it. Going into stroke, I'm sure most of you out there are familiar with stroke, but you know, it's a big issue. I mean, it's a large problem throughout the world. In the US, there's about 800,000 strokes every year. About 80% of those, or just over 80%, are ischemic strokes, and that's where we're starting with. There's less than 10% of these patients, though, in the States have the ability to be reperfused. So over 90%, there's nothing we can do for them, okay, except for rehabilitation. But globally, it is the second leading cause of death in the world. There's over 5.5 million deaths annually from stroke. You know, we started with stroke, or an acute condition, stroke and TBI, for a reason. Again, the way our product works, we don't care what's causing the issue. We're hoping that we can modulate that inflammatory component and have tissue repair outrun what's causing the issue. And that could translate into a much better quality of life. And so think about it from an acute perspective. You don't have a more extreme or hostile environment with inflammation or cell death than you do with stroke or TBI, especially stroke. So if we can show we work in that hostile environment, then we know that that opens the door to these less hostile environments for the chronic neurodegenerative diseases. Another look at crossing the blood-brain barrier. This time it's with a radioisotope that was labeled on our exosomes. And one of the things that we found really interesting here is that not only do we cross the blood-brain barrier, but we actually migrate to the infarct region for stroked animals. So we migrate to that area that's wounded. And that's what you see in the red circle on the right. That colored area is our exosomes migrating to the infarct region, where you don't see that on the left. Done multiple animal studies, as Steve mentioned. Obviously, we started with rodents. One of the first studies we did were in 12-month-old rodents, so they're middle-aged. One of the main things we were looking for there is the anti-inflammatory component. So not just local, but also systemic, because that plays a large role, especially in chronic conditions. So what this slide here is showing is beyond, you know, regulating or down-regulating the pro-inflammatory cytokines, we were also able to do that for the T cells that are involved. So we were able to show that we down-regulated the CD4 effector cells like Th1 and Th17 and upregulate, you know, the CD25 FOXP3 Rex T cells. And this particular study was also done with another exosome type control, MSC, the mesenchymal cell line of exosomes. All right, we were also able to show that we could repolarize microglia or macrophage from M1 to M2 state. Going on to another study in rodents, these are older rodents. These are 18-month-old. So obviously we can modify the immune response. We want to make sure that we can now translate that into mitigation of tissue damage, lesion size, atrophy, and then does that translate into increased function? All right, so we were able to demonstrate that. Going though from rodent to man isn't a good idea. So we wanted to choose a higher order species. And in this case here, we were looking to see if we could validate a model in pigs. And we were able to do that. But the pig brain has a lot of similarities to the human brain. All right. So one of the main things we're looking for is that gyro pattern. And you don't see that in rodents. They just don't have a gyro pattern. And when you think about the primates that we use for clinical development, they're the smaller primates, the cynomolgus or the rhesus monkeys. And as you can see, the pig brain is much larger and it has a lot more similarities to the human brain. Even when you think about the prefrontal cortex, the volume in pigs, very similar to that in man. And even the immune response is similar to that in humans. And so the next couple of slides are data from this study, and I'm going to hand it over to Steve to discuss.
Thanks, Stephen. This is a study where we, again, stroked these pigs using a permanent occlusion of the mid-cerebral artery, very massive stroke that induces. So then we took an MRI one day after the animals had a stroke. And as we've seen in the past, the control animals have really massive damage where there's edema. You can see on the left image there, a lot of swelling, inflammation, as we mentioned previously. That blue line is actually the midline, and it's a typical indication that clinicians use as an indication of stroke and how severe it is that midline shift to the left in this case. In the red arrow there, you can also see that there's a dark area that indicates that there's bleeding into the brain, as well as there's herniation. So actually tissue is herniating out of the brain. So as you can imagine, massive stroke. And in the control animals, 7 out of 8 of these animals had this very severe reaction after stroke. Contrast that to AB126 on the adjacent image here, where you can see, again, they have a stroke area. The lighter gray area indicates the animal had an ischemic stroke. But after 2 treatments, so we give treatments IV within the first 48 hours after stroke, about starting anywhere from 2 to 6 hours after stroke. And you can see that just 2 treatments here, we saw a huge amount of effect, reduced edema. The midline shift is not as acute or as strong as you see in the left side. And there's no bleeding into the brain. So there's no hemorrhagic conversion here in any of the 8 animals that were observed after AB126 treatment. Taking a follow-on MRI at 3 months later is in the animals that did survive. It's really very apparent that there's a lot of tissue that's been lost on that side where the stroke has occurred. It's amazing that these animals actually survived with this little brain tissue left. But with AB126, you can see there's still some damaged area, but there's a large amount of preserved brain tissue as well as regeneration of additional tissue to replace those dead cells in the brain. So really a massive difference. So this, from a tissue and image side, is something that we can follow in humans as well when we move into the clinic side. So this is really important and really a big indication of how AB126 works. But it's also what we were able to do, which is also important, is to get functional data. And on the left side, this is an animal walking down a gait mat where we can measure their actual hoof stride lengths and so forth. Untreated animals 6 days after stroke, this is one of the best ones, still needed to be coaxed down to the other side, even though there was food there. As you can see on the right side there, AB126 treated animals 6 days after they've completed their treatment. they're basically back to normal, back to pre-stroke levels, and their ability to move down this track, and they're really very symmetrical in this, indicating that there's very little damage that has occurred to these motor cortex of these animals. So it's real big indication, and to us, our biggest aha moment that AB126 is really a powerful anti-inflammatory and neuroprotectant agent.
All right. Thanks, Steve. So you can see from the MRI, 24 hours after the most extreme stroke, we were able to show a huge difference. That's how powerful and how fast our product works. You saw then how well that translates into function. As Steve mentioned, that pigs that have been treated only 3 times over 48 hours with our product, they were able to go back to baseline pre-stroke. So we're hoping that from showing this in the most, again, extreme environment, that translates into the less hostile environments for the chronic neurodegenerative diseases. So getting on to, we're looking to file our IND in early Q3 and start our phase 1B study afterwards. It's going to be your typical dose ascending study. All right, it'll be double blinded as well. We're going to have 3 cohorts, starting with low, then medium, then high dose. Each cohort will have a placebo. We'll have some placebo patients as well. So these are all going to be ischemic stroke patients. The way we think about dose is just the number of particles per kilogram. All right, so we're actually taking the medium and high dose from the pig study, and we're doing the same thing per kilogram in man, because those pigs were 180 to 200 pounds, so about 100 kilos. It's a safety study, but because it's in stroke patients, we'll be able to collect some efficacy endpoints as well. But it's a small end, so most of the stuff we're looking at there will be the imaging and the biomarkers. We will take the typical scales or scoring of the scales that you would do, but it's a small end, so I'm not sure if we'll have enough patients to see a huge delta there. Okay, so 3-month endpoint. We will do another 9 months of safety follow-up, but we can unmask part of the study after 3 months, so we can move on to our proof of concept studies after the 3 months is done. The main site that we're using is University of Texas in Houston. TBI, so it can be used for TBI. We've also done some preliminary work in TBI. This is also a huge issue out there right now. Just in the States alone, it's almost 60 billion healthcare costs. There's just nothing we can do for these patients, okay? And over 5 million people in the US have a disability due to TBI. So to have a drug that could work in stroke and TBI, could be a really huge opportunity for us. And to be able to have a partner that can take it beyond the US, that would be also beneficial to us. The interesting thing, regardless of whether it's TBI or stroke, once that assault has occurred, the pathophysiology is basically the same between the two diseases or conditions. One of our early studies that we did here was published online in the Journal of Neurotrauma. So it was a rat study that we did. We're looking for the same things that we saw in stroke. So one of the things that I showed you in stroke was that we saw that the exosomes migrated to the area of assault or injury. And we saw the same thing for TBI as well. Also then wanted to understand if this translates into decrease in lesion size. So that's one of the things that we were able to demonstrate. And with these photos, funny enough, they were able to get onto the cover of that month's Journal of Neurotrauma. But we were able to demonstrate in this small study statistical significance. That's how powerful the product is. We were able also to show that increased function. So not only are we showing it from the tissue point of view, but we're also showing it from a function point of view. So in stroke and in TBI. And because of that data, the NIH came back to us and awarded us another $700,000 to take it up a step to the pig model. So there is a validated pig model at the same place, University of Georgia, where one of the top veterinary neurosurgeons works. He's the one that validated the stroke model for us. He's also got a TBI model in pigs. So the NIH gave us money to take it forward into that. And this study is going to get started in Q3 of this year. I won't go through the design, but it's a large study, so we're going to get a lot of good data from that. The idea from this is to be able to show, again, the structural and the functional changes from using our product. We're going to dose it the same way we do for stroke patients. So they'll get 3 IV bolus injections over a 48-hour period. Okay. And going into the chronic. So we're starting to do the work in chronic. We started off with a MTD or a safety study, which we've just finished last month. So now we're getting started in our first chronic disease area, which will be ALS. We're doing the SOD1 model in rodents, and that's getting started as we speak. And huge opportunity for us. I think it makes a lot of sense for ALS to have a product that's as complex as ours. You can't just be a neuroprotectant or an anti-inflammatory, and you need to combine those two. And to be able to combine that plus tissue regeneration in one product, I think we're going to hopefully see the same level of progress here that we saw with the stroke patients. And then that leads us to vascular dementia as well. So being able to show that quality of life increase in dementia patients, I think it's going to be huge. And again, they're very complex diseases, multifactorial. So you need to have more than one approach. One of the other things that we're working on in the background is different ways to deliver it. So one of the routes of delivery that we've been working on in the background is intranasal. So we've also been looking at subcutaneous, but it seems to be that the data we're getting from the intranasal is really interesting. So we're moving forward with that. Again, our first formulation, though, is an IV bolus injection. Some of the other stuff we already mentioned, Steve's background, and this technology or this platform comes out of the University of Georgia's labs, which are Steve's labs. He's been working with this specific cell line for close to 20 years. It took a while for him to come up with a specific environment and cell line that produces these native exosomes that have all of these abilities to do what they do. Again, mentioned that we have a GMP facility. That's a picture of our facility right there. We've just scaled up so that we can make clinical material batches. We're going into qualification batches soon, so we can get it up on stability for our IND filing. We've got different patent families. For this first product, family 2 is the most important one. It protects our cell line, also protects anti-inflammation and treating diseases like stroke. In the US, it goes out to about 2036, and we can get an extension going out to about 2041. It's also been approved in other countries, just most recently Japan. It's still going through prosecution in China, but we hope to have that approved soon as well. And these are the milestones. So that's the story. We got a lot going on right now. And we're excited to bring on a partner that can help us with our development. So thank you. And I will stop sharing. Mark, you're on mute.
Thank you, Stephen. Thank you, Steve, for that presentation. Gathering some questions from the audience. Give me a second while we put that together. And here we go. Here's a question from the audience. As exosomes still face challenges in large-scale industrial production, the commercialization approval of exosomes is relatively complicated. How do you view the prospects for the industrialization of exosomes?
Well, it is a difficult area, and that's why we've taken it on ourselves. And so we've put a lot of effort, resources over the last 3.5 to 4 years to really make sure and fine tune our process. And the amount of process development is really large, but we've made leaps and bounds as to how pure and how much we can collect of our exosomes just even over the last 12 months, you know, as we scale up to get ready for going into the clinic. But that technique that we're using, I think it's going to be applicable for scaling up to get into the later stage. Right now, where we're at, we're fine to go through the phase 1s and proof of concepts, and we'll continue to scale up and learn how to do process development to have a more pure product as we're in the clinic in these earlier stage products. But we have the capabilities to not just get through the large registration studies, but also to do a soft commercial launch. Right? It's applicable. It's not easy to find some source or CDMO exterior or externally to do that.
Yeah, and if I could add, you know, we've always been looking at this as a scalability. Can we scale the technique that we have to make these exosomes and purify them and concentrate them in a scalable fashion? I mean, typically today in most labs commercially, they use some form of centrifugation to isolate it. We knew right from the beginning that that would never be scalable. So we moved to multiple levels of filtration and chromatography and so forth to be able to scale this, not only the manufacturing side, but also making sure that we have a clean and pure product, as clean as possible for use in the clinical arena. So it's we have a certain footprint right now, but everything we do is scalable, and just adding more to those clean rooms will increase our scale as well going forward.
Great, thank you. Another one. What is the major safety concern of exosome treatments, and how do we mitigate that risk?
Steve,
yeah, and this is actually the sweet spot for exosomes versus something like a lipid nanoparticle, where that's a foreign body. That's something that's foreign to our immune system. And of course, there's toxicity issues associated with a lipid-based nanoparticle. Now, an exosome comes from a cell. As I said through the presentation, there's a lot of advantages to that, but one of them, and one of the main ones, is the fact that the body does not recognize an exosome as a foreign entity. And so there isn't the immune reaction that's observed with other things like lipid-based nanoparticles. So we've done a number of studies, as Stephen mentioned, through toxicology and whether it's in stroked animals or normal animals. We've not seen any problem with the toxicity or immune reaction to those. So it's an intriguing area of science and research right now to understand fully why an exosome avoids the immune system extensively. We have some cues and clues, I should say, as to what it is, and it has to do with some of the surface molecules associated with an exosome that are natural and keep them from being triggering an immune response when introduced into the humans.
Okay, thank you. There's another one. Regards to the AB126, what unmet clinical needs do you think AB126 has the potential to meet in the field of neurological disease? I think you kind of went through this in your presentation. So sorry for being redundant on that question, but maybe you can just answer high level.
I think, yeah, we covered that. But again, what we're seeing today in the clinic from other companies, the larger companies, it's really not doing what we want it to do. And the benefit to patients is really minimal. And that's because these diseases are very complex and multifactorial. We're the only product out there that can address that because of how complex our product is. And that's a huge bonus to have an exosome that's made biologically by our body, because that's what it's trying to do. Our body, our brain already makes these when we have an assault or we have something going on. We're just not able to make enough to compensate for what's causing the issue. So we're helping that. We're just adding a lot more so we can overcome the problem. Okay, so again, we can be agnostic as to whatever condition where those things are happening, where there's an immune response, which is causing secondary damage or more stress on the cells, and where cells are dying, okay, mitochondrial risk or whatever's happening. And then we add in the mix tissue regeneration.
I guess with regards, and I think you might touch upon this, but I think they've been a little more specific in terms of your future development plans for AB126, sort of next 3, 6, 9 months milestones, catalysts that we should expect?
Yeah, so with the native exosome, which is our first product, there's going to be lots of milestones, because again, if we can have this agnostic approach, then we can just keep building a larger pipeline, assuming that it's working in the clinic for the acute and the chronic conditions, and we can take it wherever we want. But some of the fun and exciting stuff in the background is exosomes can be used as carriers. So we're actually doing some cargo loading in the background to learn the best ways to do that. And we've done some work with loading biologics and small molecules. And so our next product beyond AB126 could be a combination product with AB126 combined with another therapeutic.
Another one, can we expect the same effect of AB126 for both ischemic and hemorrhagic strokes?
Steve, do you have an answer?
Yeah, I mean, that's an interesting question. Of course, hemorrhagic strokes are really devastating. It affects anywhere from 10 to 20% of all the strokes are hemorrhagic, and there's no treatment for those as well. As I mentioned, there is innate ability of exosomes to have effect on vascular cells, on the endothelial cells and the smooth muscle cells that are surrounded in the arterial function. So we believe that it'll make the brain less leaky and less likely to have that hemorrhagic stroke to start with. Repairing after a hemorrhagic stroke, having that same ability to make the capillary cells and other portions of the vascular system less leaky is an innate opportunity for us. But of course, the biggest problem is ischemic stroke. That's what we'll tackle first. But there is an opportunity for hemorrhagic as well in the future.
Great. A few more. Based on preclinical studies for acute ischemic stroke, does the exosome treatment have a treatment time window similar to TPA?
And to answer that question, we can go beyond the TPA window. There is a reason why you can only administer TPA and has some very adverse events associated with TPA and some indications. So you want to narrow that window. We can go beyond. So if a patient presents themselves to the hospital outside the window of TPA, we believe that AB126 will be able to be used to help those patients as well. And I should mention, just as a follow-on to that, is that, there are, increasingly, there's being used intervention to pull out the blood clot from these major arteries. That also induces vascular damage and can impair blood flow, and could increase the chances of a hemorrhagic stroke happening at a small level. So we think that AB126 could actually act as a protective in combination with a what was called a thrombectomy, or mechanically removal of that blood clot. So I think what I'd like to say is that there's an opportunity to combine these treatments. They're not acting in the same way, so there's perfect opportunity to combine treatments and have a beneficial effect beyond the TPA or thrombectomies.
Mm-hmm.
And Mark, I'll just make a comment on that.
Once
we get the safety study done, that's why I mentioned, and you can see it on one of our slides, multiple proof of concept studies, because once that safety study is done and we have an open IND, we can be trying different things for stroke patients. And one of them, at the beginning, we're doing our first treatment within 6 hours, but we believe it'll still have a beneficial effect if we're way outside of that. And so that's a study that we can also take on as a small proof of concept. We can push the boundaries more and more with different populations, within the stroke area. And it would be exciting to try that.
I think we have time for one more. And I believe you kind of touched upon it, so maybe we can just elaborate a little more. But since AB126 has a potential for treating neurodegenerative conditions, do we have plans to extend the indication to diseases such as PD and AD? And I think you touched upon that, but maybe you can elaborate more in terms of timeline or sort of, what are the corporate goals here?
Yeah, so I mean, the first one's to get these two done, you know, one in each of these categories. And once those are done, it's really about the resources. You know, the resources are going to limit us as to how quickly and how far deep we can expand our pipeline. But there's no reason why this shouldn't be applicable to all forms of dementia, not just vascular dementia and motor, you know, neuron diseases. I mean, there's no reason why Parkinson's couldn't be part of our pipeline. We just don't. I mean, we're a private company. We have limited resources, so we're starting where it, I think, makes the most sense with ALS and vascular dementia. But it's, you know, it's just a matter of getting the resources to expand that.
Great. Great. So any closing remarks to our global audience?
I just want to say, I don't believe there's any other type of product in the world that can do what ours does. And so there's a lot of people that are waiting to understand if this does translate into the clinic. But here's the thing, we believe it will because of the robustness of our preclinical package, especially that porcine model. If this does work in the clinic, and the opportunity here is huge, there's a huge amount of diseases that could be treated, or conditions that could be treated with this, and they could be used alongside or adjunctive to what Alzheimer's patients are getting today, or Parkinson's patients are getting today. There's no reason why it couldn't. All right, so it's a huge opportunity.
Great, thank you.
And I just want to thank you, Mark, for hosting this and the opportunity to present what we think is really exciting opportunity to treat a number of patients in the future.
Of course. Pleasure's all ours. Thank you. Thank you, Stephen and Steve, for your knowledge here on your exciting scientific achievements, and look forward to Aruna's future catalyst and milestone achievements, advancements. So that concludes our MSQ webinar. Thank you for attending.
All right. Thanks, Mark. Thank you, everyone.
Thank you, everyone.